Okay, so we were here last time on this slide and the slide after that. So essentially, we talked about attenuation control, and one of the examples that I'd given was, looking at how the trick operon works, and usually it's similar for all kinds of amino acids. And we will continue from here because we talked about three kinds of control: positive control, negative control, and the attenuation control in the synthesis uh, of uh, of various. I mean, the regulation of genes and synthesis of proteins from genes. All right, so. Um, okay, so now let me go down from here. And this is essentially what I was telling you earlier, that we have three levels or four levels of information that gets moving around from the gene to the mRNA, from the mRNA to the protein, and from the protein to the metabolites. And to represent that, we have we'll have one slide later on, but the whole purpose was here that we would talk about this one unit, which was the primary subunit of the entire transcription network that we're going to look at, right? And we have two parts to it. One part is this general part that I'm going to talk about, and the other one is going deeper into this and looking at how this whole thing works. So, and the last question that we ended up in the previous lecture was that, we were discussing, where is it that you draw the boundary? And I said that, that we have to look at the problem that we want to address. How are we framing the problem? The question of the problem will tell us how deep we need to go. And for us, at least the way we are doing this course right now, it's important for us to know how to do at least this one subunit, which is to look at how this transcription regulation works. And the way the transcription regulation works is that you'll have, maybe in this case, the way I've shown you the example here, is that you have a protein that regulates the synthesis of this gene. So you can have an inducer, uh, and that inducer will act on this regulator protein and then help it to induce the synthesis of this gene. And the way this, if you were to monitor this by a fluorescence as a protein is getting synthesized, let's say the protein has a GFP tag, then you'll see that the fluorescence will go up in this kind of a signal that we see here. And we talked about these operator sites and the promoter sites earlier also and how they work. At least we talked in detail about the lac operon and the arabinose operon. Now, from here, we need to go and look at what I told you earlier is this four level of information that we essentially have. And just even if there was just a single protein being synthesized, you can see how complex it could get if all the possible connections that are allowed were permitted to happen just on that regulatory control. Obviously, it never happens like this. It's just showing you the entire set of information that is there. And you can see from here, you can infer at least that this could be quite complex, right? And that is the reason why that you need to be very careful on how you do the modeling, what is the information that you use, and how far you go into the depth of, let's say, a single subunit. And I talked about multi-layers also, where you can go to the depth of each subunit, but then when you're doing the total modeling and the total simulation of the system, then according to your needs, you cater to what needs to be dampened in terms of the graininess of each layer. So you could have one layer which, although it is very refined and has a very fine uh, resolution, you could make it grainy because at that point of time you don't want it to kind of uh, you won't want to look at the details that are there at that layer. And one such example I will give you and show you how detailed it can get when I talk about this level, which is going from the gene to the mRNA to the proteins. I'll just show you that one example in the next few slides. There is a paper on which I'm basing the next few slides, and I'm using this example very specifically because this example is worked out and it is given in a very nice detailed fashion. And if you read this example, you will get a fair idea of how this thing happens. So that is the reason I'm picking up this example. It came up in mathematical biosciences. All right. before we go to that, there's one other layer which is here in this level, and that layer uh, is or is in this sense. If I were to just look at a sequence of these events without any connections and feedbacks, then I can see that I could write if there's an enzymatic connection, and if one substrate is giving you one product, then you can get to this point, right? And if this becomes a substrate for the next level, then you can get another set of equations which are just simple mechanical kind of equation that can give to this point. But obviously, from here to here and then to there, you can see, and you've already done this, maybe, in one or two examples that we had done in the class and in the assignment, that this gets quite, and just to get those values and put it there, it's quite a lot of work. Just imagine if, along with this, you had some feedback. Let's say if I was looking at these two layers and then now I have a feedback, let's say from this gene going back to the signaling molecule to tell, maybe through some other route. I mean, it need not necessarily be going back directly to the signaling molecule, but to something that initiates a signaling molecule. And then that is captured here in this feedback. And of course, it will be much more complex than this. But as you can see, if I were to allow this, then obviously, depending on the sign that is here, as well as other things that can happen in terms of the magnitudes of these values here, I could see that this could get quite complicated, right? And just writing out the equation for this will become difficult. So how many of you know how to write out the equation? For example, if I asked you what is the relationship or the output x3 based on changes that happen in x11, how would you be able to do this? Would you be able to write out the transfer function in this case that transfers the?
Yes, sir.
Okay.
Yes, sir.
Can you tell me briefly how it works?
So for the first one, it will just be since like x1 transfers to x2, so it will be we can take v1. So just a second.
It'll just be one into concatenated one, right? Your x2 will be added from here, right? So that's all.
Yes, sir. Yes,
so,
sir.
So you can just write x3, but of course this is, you have to keep in mind, this is nonlinear.
So
you will not be
able to. X2 is the product, right? Here.
Yeah,
so dx2 by dt is equal to v1, right?
Yeah. So
but we'll integrate that and
yeah, there is a dependence here. So unlike if you had a linear system where you could cancel out numerators and denominators, here that will not happen. Just keep that in mind. But the principle remains the same. That means forward divided by the loop, right? So in this direction, it will be the forward. So if I were to write this as G1 and G2, then I'd say G1, G2 divided by the loop. Then I'll have to work out how much this is. There'll be there'll be G1, G3 by 1 plus G1, G3, and then a G2. So G1, G2, G3 divided by, and then you work out the entire loop, and then that will give you how this works out. Depending on the sign, it will change. The signs at the bottom, right? Do you remember that part? How this happens? If not, just go back and see. But
yes.
But it's not really that important because we're not going to work out, because there are two ways to work out. If you want to work it out in the normal fashion, you have an idea. But otherwise, in any case, if you have the set of equations and you have the initial conditions, then you can go back and integrate numerically, get the answer. Okay. Now I'll bring out that. Oh, okay. I think the example comes later. I'm so excited about the example that I keep saying this. But let's look at these things one by one. And this representation I'm showing you here, I'm going to come back to this again in a subsequent lecture. Okay. So there are two things that I had said earlier, and you'll see when I come to that point, you will notice that I've talked about this earlier, and then you can go from there to see how this whole thing works. Okay. Now there are many ways in which you can represent this thing. So what are the steps? The first step is that you get what is called a wiring diagram, right? And based on the wiring diagram, you essentially will have how the information flows. Now here, I'm showing you the breakup within the wiring diagram. That means if you look at this, for example, these connections which are in blue and red are showing you how the information blue, red, and black. The inside part is, of course, the dotted line. So this is the direct information flow that is taking place. So you can see that there is a negative feedback coming here from C. There is a positive going from D, and there is a transcription that takes place between A and B, and then these two together again give you another feedback here. So in the system of A, B, C, D, we can see that they are all interrelated now, right? And the directed graph, which we'll do later on, is shown like this. So if I were to do a directed graph, then A has its own self uh, uh, information going back on its own self. So it's a reflexive condition that happens. A can then produce C because there is a combination with B. So then the C will go to D and D will go to B, and B and of course A together will then give you C, right? So there's a correlation that happens. C goes to B and B goes to C, but that self thing, that feedback is here, that gives you this information which is happening here. All right. Now you look at the Bayesian networks. It essentially tells you that A will give you C, B will also give you C, and then C will give you D, which is the final thing that is coming out here. So that's the way this would work out, because condition dependent. So A has to first form C, B has to form independently and then come to C, and then together they would give you D. That is a condition. If you were to do Boolean networks, it's something similar to this one, but there you actually would include the signature of that edge. That means whether it's negative, inhibitory, or positive, activating. So that's how this works out. All right. So these are, yeah, and there has been an attempt to essentially generalize the way in which you would put these uh, components. That means if you have an mRNA, then you are using an elliptical shape. If you're looking at a gene, then you're writing it in a rectangle. And if you're looking at a protein, then you're putting it into a hexagonal box. So these kind of shapes and colors and signs and dotted edges and bars, all of these can actually give you understanding of uh, or coding the wiring diagram. And if you go back to the Institute of Systems Biology, from where initially uh, uh, this whole thing started, they had tried to do this, but I think people have generally not stuck to this kind of a protocol, and so that has given some kind of an difficulty that arose later on. But anyway, it doesn't really matter. From the context you can pick up, and we'll talk about each of these in subsequent lectures. But primarily, what we're going to talk about is, right now in this case, is looking at ordinary differential equations to discuss and to understand this basic subunit which is there. That is, how is the gene regulation taking place? All right.
Sir,
So yeah, Ratnesh.
can you please describe what is Bayesian network, sir? I do not know.
Okay. So Bayesian networks is something that is based on probability. Okay. So Bayesian networks means there is a conditional probability which takes into account an event that has taken place, which influences another event which is going to happen. Right. So now if you look at what we're trying to do is we're trying to understand how D is going to form given that something is initiated from A. Right. Now there is a certain probability that A will happen. There is a certain probability that B will happen. Once A and B has formed, there is a certain probability that C will happen, and that is conditional. And that is where the Bayesian network comes in. That means you are actually having conditional probabilities to define these things. So now, how you choose this probability distribution function for each of these cases will depend upon what you infer from experimental data that is there in the literature, or from experimental data that you generate based on maybe certain, because you can't really generate all the density function and other things that may be required, or the probability that may be required, just by simulation, but by itself, but or by experiments by themselves. They may be biased. So what you do is you essentially find the parameters, then simulate it under various conditions, and then from there infer what should be the probabilities that are there. Now once C has formed, and you can see here, then the condition that D is synthesized just depends only upon C. But of course, if you go back and look at it, then of course C is dependent on A and B. So therefore, the synthesis of D will depend upon the probability of A and B and the conditional probability of C forming, given that A and B have formed. Clear?
Yes, sir.
All right. And this is the primary equation that you have to know and understand. And this is maybe the most fundamental equation for transcription networks. Okay. and this is based upon the Hill's equation, and the Hill's equation is given here. As you can see, that a certain output Z depends upon some. This is for biological systems that have been specially adapted. Depends upon some kind of latent or basal synthesis that takes place, some amount, and then there is the synthesis that is dependent upon some kind of an initiator S, and there is a certain amount of degradation that take place always, because because these being real systems, they cannot be infinitely producing. There will be always some kind of degradation term, so that there are two opposing effects which balance each other in the long run. And from this, if you put the steady state condition, you just put left hand side equal to 0 and make Z the substitute subject to the formula, you can get the ZSS will become, and you can see very easily that that this will work out to be giving you this value here, right? Where A is taken inside, and A, as you can see, is something like this. So it's very simple, not very difficult at all. And the condition that you find here for repression and activation depends on this coefficient mu here will be either 0. If it is 0, then obviously the numerator will become 1, and that will give you this automatic reverse switch. That means from high it go down to the low value, like that. And this will be the if it is equal to 1, it is the positive switch that is giving the activation going from 0 to higher value, some steady state value. Then there is this value n, which is called the Hill coefficient, right? So K is the Hill constant, and this is the Hill coefficient. So K is Hill constant, keep that in mind, and n is Hill coefficient. Now the Hill coefficient depends upon how the degree of cooperativity that is there at which allows the subunits to essentially come together and support each other as the signal develops. So that can happen in one way, or what is the total number of subunits that are formed together that are actually activating this conversion of or rather the initiation of this Z from some signal S. So that is, so usually it will be in the range of 3 to 4 if it is a normal system, but people report fractional values, people report very small values. If it is 1, then it becomes a Michaelis-Menten equation. So keep those things in mind. And then of course we have the value again, which is the same meaning as we have for the Michaelis-Menten coefficient. This is the half value. That means if at some point this is half the value of the maximum value of that it can take, then this will be this K value is that value at that S value, which is, or Z value which is there, the maximum Z value, half of that, and the coefficients will be nearly the same. The units will be the same, S and K. So keep these things in mind, and of course, as you can see here, the parameters K prime or K are signal independent and signal dependent. So there are two different things here. So K prime is not depending on the signal because it is a basal level of synthesis, whereas this depends on this because now you're looking at something that has gone into this system where it is now depending upon this KD that is coming here. Okay, so this KD will actually take away some values of the signal, so therefore it becomes dependent on the signal in the other sense. All right. Now this is also a very important slide, and you have to keep in mind that there are time scales that are there, and we'll see why these time scales are so important. So when I come to looking at these transcription networks in detail in the course of this lecture that I'm giving now, you will see that these time scales become very important because based on these time scales, the cell is able to generate different kinds of profiles, which are understood by the cell as either a signal or as a change in the concentration or as an impulse or a trigger. So those kinds of things are based on this time scales, and evolution has fixed this. And it has been my experience that given a system, suppose I pick up for E. coli, and I build up a system of let's say 10, 15 genes with all the regulation together, and I pick up all the values that are there for one species, then generally, I'll get a fairly good response uh, for that system if I were looking at, or a consistent responses. That means what I mean to say is I'll have the simulation converge in a nice way. I'll also have uh, the responses that I'm expecting. Let's say if I were expecting oscillation, I see that also. But if I kind of mix or I start changing and varying one of those parameters, which have been tuned for the entire system, then I find difficulties in either converging or I'll find inconsistencies in the response that I see. And there is no way that you can fix it really, except that you actually go back and use values which are close to those, within 10% of those values of the parameters. Else it sometimes they'll just give you an anomalous result. So keep that in mind. So now with that as a background information, just look at these 4 points that I've stated here. So the input signal for transcription activities is in the sub-second levels, in the milliseconds. If you're looking at binding equilibration, then it's in the seconds level. If you're looking at transcription and translation, then in the order of minutes up to 5 minutes. And if you're looking at protein products which are going to happen, then that takes from minutes to hours. And sometimes maximum on the order of hours for one cell generation to take place. And about half an hour, 30 minutes is for a faster growing organism like E. coli or maybe some of those other organisms which are there. Okay, so keeping that in mind, now I'm going to go to this example. And this example is there in this paper, and I'll just exit from here and I'll come back to that paper, which I'm going to show you, and I'll come back to that, come back to this presentation after that once again. So the paper is here. I've kind of taken the liberty of actually looking at a few of these things here. So can you see the paper, all of you?
Yes, sir.
Yes, sir.
So this is a maths bioscience paper, and this is a single author paper. And you can see that this is quite old now compared to what we are doing. But it is at the time when these were things that were being investigated. And this is the analysis of protein synthesis dynamic model in eukaryotic systems. And they are trying to understand how the inputs can control this. Okay. And this paper I will post so that you can take a look at it. But now we will go back and discuss this paper in the lecture now. So I'm going to kind of close that. Cancel, not that. I can go back here. And I'm going to stop that and I go back to my presentation. So give me a second. Oops. All right, so we are back here now, and let me just talk about this a bit. Okay, now look at this problem. The problem is nothing but they're just trying to model one thing. They're trying to model how the methynyl initiator affects this synthesis of protein. So they're looking at, so when you're looking at eukaryotic systems, they have what is called a eukaryotic initiation factor, EIF2. And this is, and bound to GTP, and then only this initiation process starts. There's also a GCN2 unit, which is the general control non-derepressible 2 factor, which is also required in this process. So these two are the main thing. So the question they are asking is, if we know that this level of EIF2 is actually upregulating or controlling the synthesis or the transcription rate or the translation rate here, then can we control the concentration in such a fashion that we get the desired rate of translation, right? Now recall, in that problem that I was talking to you about about methionine, that I give you as an example, that big model that I showed you. In that model, what I was asking was that, what happens to the tRNA if we have upstream disturbances in the signal of aspartate, right? And what we found is that, as long as, the synthesis is actually committed, you will not see any change in the synthesis of the tRNAs of methionine, right? So that is a very big factor, because there is an energetics that is linked to this, and here also we see that energetics is linked to it, right? So the ATP ADP subunits along with the GTP GDP subunits will actually uh, affect. So I mean the bound subunits with this exchange of these energy currencies will actually control how exactly this happens. All right, now let's look at this. So now you have to look at this carefully. Yeah? So this is a little uh, uh, unusual, because we are going to go into the details of how this whole wiring diagram is done. So try to understand this from the physical point of view. Forget about these equations right now. Just look at this figure. So in the figure, what you see is there's a methionine tRNA, which means that this is the methionine RNA, because you're looking at the initiation step. So it's not going to be the methionine tRNA, but it will be the methionine tRNA, because that's going to be the first step. And what you see is that there is a EIF2 GTP. That means there's a factor which comes here and joins. Then that gives you, and we'll see what this TC factor is in the next page. That gives you an intermediate. And that intermediate, so this is that bound intermediate. And then that is actually going to be the ribosome 40 subunit, 40S subunit is going to bind here. And that's going to give you another subunit. And then you have a control that comes from hormonal side. And then that EIF4F is going to be binding to this, and then that gives you. So these two are the 43S and the 48 subunits. So there is a modification that takes place, and those subunits are here, and then this will bind with the 60S subunit to give you the 80S subunit, and that begins the elongation process. Okay. Now there is this uncharged tRNA which comes back here. There is this charge, uncharged, I mean in this D, so that part comes back here. You also have the uncharged EIF2 come back here, and then this control takes place, and then that goes back here. Now the control has been shown in details here. And here again, you find that there is this GCN2 factor here, right? So you can see the GCN2 factor here. I'm not getting the, okay, never mind. So just look at this. So there is this factor here, which is the tRNA. Then that is going to be working with, so this is the input that comes in. Then there you have the E2, which is, I'll show you what that is again. And then this control goes further inside, and you can see everywhere I'm showing you how this EIF is getting controlled. So this ET is actually this EIF that is going outward. So how is that getting controlled is shown in detail here. And you can see that you can go and start blowing up. So this is exactly what I was telling you earlier in the previous class, that as you go further and further or deeper into it, you can actually keep on blowing up the degree of resolution you want to see what's happening. And it is not necessarily required in everything that we are doing. So at the level which I was showing you in the methionine problem, I did not look at all of this. I only looked at just the functions that were there in terms of kinetic equations that were controlling or that were defining the rates of transcription and translation. And in that, there was this factor that the enzyme is getting synthesized, and so there's a rate of synthesis of the enzyme. So that was the part I used. But here you see that you're looking at even protein, at the level of proteins, you're looking at how these concentrations are changing. Now let's come to this part here, this equation. This equation is something that is very general. Okay. Now, if there are students here from mathematics department and from physics department, if they are here, then they'll recognize this as the most general form of writing a nonlinear equation. So, you have a state which is changing with respect to time, and that is dependent upon time, that is dependent upon the states, and that is dependent upon the inputs that are there in the process. And what you're saying is that, well, this is an n dimension. That means there are n components of this vector of the states. There are m components of the vector of the inputs, and then this is what you're measuring. So what you measure is on the outside, and that is also a function of the state, because unless you have a state, you can't measure it. And this is nothing but showing you the instrument. So the instrument is telling you, well, there is a certain way that you convert. So for example, if you had a length of mercury, with temperature, the length increases, but the length can be mapped to temperature. So that conversion is this H function. So that is, and for each measurement that you make, there is a function like this. And if you have P measurements that are being made, then you have, this is going to be of the order of P. So that's the reason why it's made like this. So there's nothing more to this. It's in words. This is very simple to understand. Okay. All right. Now, any questions?
No, sir.
Okay. So what I'm doing here is now explaining. So this, of course, you have to sit with it. So this is an illustrative problem. So I'm explaining this to you, and when you go back and look at it, you just sit with it, and you'll see that we have E2 here. So ET is this complex that I told you about, which is coming out from here. E2 is the EF2 complex, which is there. That is shown inside, should be here somewhere. So it's not shown here, but it comes as EB is shown here, of course. You can see that EB is there. EPD, which is this complex with the GTP, that is also shown here. You can see all of these different. So GTC is this one, like I told you, and TC is here, which is the ternary complex, like I told you. Then the 43 subunit, and then the 48 subunit, which is there. Clear? So now, if I were to go back and I put rate equations for this, just like I showed you earlier, how you can write it, you could write these equations out. So this is not really a big deal. The only thing you have to keep in mind is that you have a way to make the notation clear. So 7 to 1, it means that 7th to 1, reaction. 2 to 1, where is it coming from? So if you go back here, you will see that EB, there are two components to it. There'll be one which is getting used. So this is one which is getting used. There's another one that's getting synthesized. So if you write out those equations, so you will be able to write this out. So this comes from the physical translation of the physical problem into this part, and you can go one by one. So each of these will, the dot means it's a time differentiation. So dd dt would be dE b dt would be this E dot b, right? So, and you can write out these equations. There are, in this case, there are 9 such equations written out. So it doesn't matter. So these are things that you can always handle, right? You can go to first principles and you can work it out. And you can, sometimes there are some models that are there in the literature, you can use that. That's not a problem. So you can see that if I were to look at each of these, I could actually go back and map it. So I'm just showing you another example in case you're not able to visualize it, then it's very easy to see from here that, for example, if I'll take a simple case, let's say AC. So AC will be 5, 2. So this must be step 5, and that must be, maybe this is step 5, and that could be step 2. So 5, 2 is the rate. So AC is that rate at which is getting removed for that case. And then you have another, the rate at which TC is going to synthesize this, and that is dependent upon 5, 1. So I don't know, maybe some other way of notating here. We have to go back and look. But anyway, you can have a logic for noting this, or maybe it's a 5th equation or some other notation that is there. Anyway, so there is a unique way of numbering this, and you could essentially go back. So this is 5, 1, the 5th reaction, 5, 1, 5, 2, 5, 3. That's the way it's done. All right. So there would be a nomenclature for this, and then but you can clearly see that there's one rate that is synthesizing it, one rate that is removing it, and that is getting captured here. It's very clearly shown. All right. Now, so this is again, the breakup. So when that first equation that I told you, where everything is combined together, that usually can be broken up into what's called a kinetic term and a transport term. The transport term here is shown as separately because now you have what's called an input. So eta is going to be something that actually impinges on the system and then changes the rate at which this changes. Okay. Now it's going to be difficult if you have this set of equations and then you try to do the analysis. So one way to take care of this is to linearize it. How do you linearize? Use Taylor's theorem like I showed you earlier. So you look at some condition that is useful to you, or some condition that is a steady state condition, and then based on that, around that point, you start differentiating everything. You take partial derivatives, evaluate at that point, and then take the linear translation of that. Okay. So this I could have got this term. Uh, let's say look at this one. So this is linear. Let's look at this one. The d dot. Then this, if I were to partial differentiate this with respect to with respect to EV, then I'll have K21 EPD, and I would have to evaluate this at EB0 EPD0 times EB minus EB0, right? So that would be the way I would do this, and I could go ahead and do that here also, right? I hope if you want me, I'll latch on my writing pad and describe this. Is it okay if I proceed, or you want me to go through that derivation a little bit?
So could you just, like, in brief, could you just explain?
Okay, I'll do that. Hold on.
Yes, sir. It will be better if you can explain, sir.
Okay. Let me see if I can get the pen here. Pointers. Let me see if the pen works here. Then does it work the way I want? No, I don't have the control. So I'm going to Control Z here. Just give me a second. What I'm going to do is escape. Yeah, just give me a second. Okay. So let me. So maybe I'll stop the recording for a minute because there'll be a gap here otherwise. I'll come back to